Metabolism is basically the sum of all the chemical reactions that occur in your body. Reactions that occur in your body involve enzymes. In enzymatic reactions, the reactants are called substrates and they are converted into the products. The substrate will bind to the enzyme, and the enzyme will help the substrate to be converted into the product. Chemical reactions that occur in your body are called biochemical reactions, and they typically occur within pathways. In other words, you don't usually have a single reaction occurring in your body, but what happens is you have multiple reactions within one biochemical pathway. We'll look at this later. This is an illustration of an enzyme. The enzyme is the purplish molecule here. Enzymes will be proteins, and their function is to speed up chemical reactions. In other words, they are an organic catalyst. The substrate will fit into a pocket on the protein, the enzyme, called an active site. Once the substrate is bound to the active site, it will then be converted into a product, and the product will be released. The enzyme then can be used again. So enzymes are used over and over again. As mentioned before, the chemical pathways in your body require enzymes. Remember, enzymes are organic catalysts, which means that they speed up chemical reactions. They are unchanged in the reaction and they can be used over and over again. So you don't need a lot of enzymes for each one of your pathways. The way that an enzyme speeds up a chemical reaction is by lowering the amount of activation energy required to get that reaction to start. Turning an enzyme on or off will determine which reactions will occur. So how do you turn enzymes on and off? Your body uses what's called competitive and or non-competitive inhibition. Let's take a look at these. Remember that substrates bind to active sites on the enzyme. Once the binding of the substrate to the active site occurs, the substrate is converted into the product and released. In non-competitive inhibition, a second binding site exists. It's called an allosteric site. An inhibitor, in this case represented by the blue ball, can bind to the allosteric site, and when it does that, it changes the shape of the active site. If you change the shape of the active site, the substrate can't bind, therefore the reaction will not occur, and the product won't be formed. Competitive inhibition involves another molecule that actually competes for the active site with the substrate. If the inhibitor binds to the active site, the product will not be formed. What determines whether the inhibitor or the substrate binds to the active site is the concentration. The molecule in the highest concentration tends to bind more often. So let's review. This is a linear pathway. You can see that A is the first substrate and it's going to be converted into B, and B then becomes the substrate for the next reaction. In this illustration, the E stands for the enzyme, so each reaction is going to have its own enzyme. Remember that the end product of this particular pathway is E. That end product then can be an inhibitor. If you're producing more E than you need, or if you've produced enough of it, you don't want to continue producing it. So that end product then functions as an inhibitor by going back to the enzyme in the very first step or reaction of this pathway, binding to its allosteric site, and inhibiting it. By doing that, you turn the whole pathway off and you're no longer producing the product E. There are different types of chemical reactions. Anabolism involves taking smaller molecules and combining them to make larger molecules. Catabolism is taking larger molecules and breaking them down into smaller molecules. For example, when fructose and glucose combine to make sucrose, that is anabolism. If you take the sucrose and break it back down into glucose and fructose, that's catabolism. Redox reactions involve reduction and oxidation. The metabolic pathways that you will be studying will involve redox reactions. I encourage you to remember the saying, Leo goes Ger, where Leo stands for loss of electrons, oxidation. When a molecule loses electrons, it has been oxidized. When it gains electrons, it has been reduced, and that's the Ger. In redox reactions, remember you have reduction and oxidation occurring. Let's start with reduction. Recall that in reduction, a molecule gains electrons. We can follow those electrons by following hydrogen ions. So you can tell if a molecule has been reduced by looking at the hydrogen ions. If you see hydrogen ions that have been attached or bound to that molecule is being reduced. So if you look at the reaction of pyruvate going into lactate, pyruvate is being reduced into lactate, and you can see hydrogen ions being bound to the pyruvate to convert it into lactate. In addition, a loss of oxygen is also reduction. Remember, when hemoglobin unloads its oxygen, it becomes reduced. Oxidation, recall, is the loss of electrons, Leo. Again, we can follow those electrons by looking at the hydrogen ions. So if you look at the NADH plus the H encircled by the green, you'll notice that it loses its electrons and it's lost in pairs, and so NADH becomes oxidized into NAD. In addition, oxidation can be the gain of oxygen onto a molecule. Coenzymes are going to be used to transport those electrons. And coenzymes, the ones that we're concerned about, the ones that will be transferring electrons from one reaction to the other, are derived from B vitamins. And the two that you will need to know are NAD, nicotinamide adenine dinucleotide, and FAD, flavin adenine dinucleotide. This slide here just shows you reduction and oxidation occurring. So what we're going to do is we're going to look at the first molecule there with the 4 hydrogens. When you go from the 4 hydrogens over to the 6 hydrogens, you have gained electrons. Remember, we're following the hydrogens, and so reduction has occurred. So that molecule, the 4 hydrogen molecule, is reduced to the 6 hydrogen molecule. In the opposite direction, remember Leo, you're losing electrons, so you go from the 6 over on the right to the 4 hydrogens, so that is the loss of electrons. Remember, just follow the hydrogens. You can also follow the oxygens. So if you look at the bottom reaction, start with the one on the left. You see the oxygen molecule there? Now what we're going to do is we're going to remove that oxygen molecule as the reaction proceeds to the right, and that would be reduction. If the reaction goes to the left, you're actually adding the oxygen molecule, and that's oxidation. There are going to be some terms that we'll be using as we study the metabolic pathways. So I encourage you to go ahead and learn the definitions of these terms. Phosphorylation involves adding a phosphate group onto a molecule, and the enzyme that does this is called a kinase. Dephosphorylation is the opposite, where you're removing a phosphate group from a molecule, and the enzyme involved in this type of reaction is a phosphatase. If you look at this reaction here that I've highlighted in yellow, look at the protein. So when the protein has a phosphate added to it, phosphorylation has occurred, and you can see the enzyme kinase there for the phosphorylation. Now, if you take the protein with the phosphate attached to it and remove that phosphate, that is the dephosphorylation occurring. Decarboxylation involves the removing of a carbon dioxide, and the enzyme involved in that is decarboxylase. Dehydrogenation is removing hydrogens, and the enzyme for that is dehydrogenase.